It's a long time. So,
Yeah. Can we start? Okay, good morning, everybody.
Morning.
Very glad to see you all here. And also, I would like to welcome you to our very first lecture in this Department of Plant Protection, because this is the first day of the semester. So I just looking around, and I think I only know some faces familiar to me. So it means that most of you are new students in this department. So welcome. And you are very lucky because today we have a special speaker for the lecture in our department. Today we have two speakers actually. The first one is Dr. Sota Koeda. Dr. Sota Koeda, I know you have read the flyer. He is an associate professor in the horticulture science lab at Kindai University in Nara, Japan. This is the first time I met him, but I've been in research collaboration with him through the Green Project. So I also would like to introduce the researchers from Green, Dr. Kusuma. She is working with Dr. Koeda and then with our team. Yeah, we work for the Begomovirus Resistance in chili pepper. So I'm very excited to meet Dr. Koeda today and to hear his lecture. For the second speaker, this is also very exciting because she is a very young scientist, researcher. It's always exciting for me to know there is a researcher or scientist working with plant pathology or agriculture. I would like to welcome Dr. Ph.D. candidate, Nadya Syafira Pohan. So she is a Ph.D. student under the supervision of Dr. Sota Koeda in Kindai University. So she also would like to share some of her research data, and probably you can give some, sharing your experience during your study in Japan. Probably some of them would like to follow your path to continue the PhD abroad. Yeah. So without any further, I would like to welcome Dr. Sota Koeda.
Sebentar. Oh, sorry. Ingat dari saya aja. Next.
okay. So, good morning, everyone. I'm very much pleased and honored to be here in Bogor, IPB. Thank you very much for that. Actually, we have been connected with Indonesia for quite a long time, like more than 10 years. So I'm going to introduce our recent research activity related to this topic. Next one. Okay. So I was born in Kyoto, Japan in 1982. So actually yesterday it was my birthday. I became 43. I'm already mid-age scientist. Thank you. And I took my bachelor, master, and PhD in Kyoto University, which is a national university in Japan. And my major is basically horticulture science, but gradually I'm using Begomovirus. I'm interested in virus also. So I'm applied research. I prefer applied research myself related to vegetable breeding and horticulture, breeding science, and biology. Those three are my keywords. Okay, let's go to the next slide. Okay, so I've been experienced three laboratories. The first one in Kyoto University, I stayed there as a student for quite a long time. And in 2011, I got my PhD. And I was really lucky that I had a position, permanent position in the experimental farm in Kyoto University as an assistant professor. So then I became a PI, and I spent more or less 3 years, and then moved to Kindai University from 2015. So already spent like 10 years in my career in Kindai. Kindai is a private university, and most probably I will become a full professor from next year. So, and since my bachelor study, I've been using pepper abstract. So my main plant species for research is pepper. Okay, so please go to the next slide. Maybe you may know Japan. We have Tokyo, the capital here, and our area is in the central area, Kansai. I've been in Kyoto and then moved to Osaka with my previous experimental farm in Kyoto University, and then I'm staying in Nara. Nara is the oldest capital of Japan. Before, when Kyoto was capital, the capital was in Nara. Okay, please go to the next slide. Just to introduce our university, Kindai is one of the biggest private universities in Japan. And especially we are very strong in the area of agriculture. And not only agronomy, fishery is very famous because we are one of the pioneers to culture the tuna. And we have 6 campuses. Main one is in Osaka, and our faculty of agriculture is located in Nara. Okay, let's go to the right. And since 2010 or 11, we have been published more or less 40 papers. And I checked this yesterday. More about half is Q1, half is Q2, and some from Q3 journals. So yeah, I think we are working really nice. Okay, so please go to the next slide. And international collaboration is always my interest. And Indonesia has been the most important collaborator. Because of the Professor S.K. she also took PhD in Kyoto University, in the same laboratory as I did. So she was in PhD when I was undergraduate student. So we get connected. She's like my elder sister. And we have been collaborating related to Begomovirus research since 2010. Okay. And several years ago, I had the chance to do my sabbatical in Spain. Spain has also a big program with Begomovirus, especially in tomato and cucurbit. And my boss was Enrique Moriones. He is famous for tomato infecting Begomovirus. He has already retired 2 years ago. And there was another professor, Haru Papu from US. He was doing a sabbatical also. So he became a good friend of me. Okay. Okay, he's very nice guy. Okay, please go to the next slide. What we did is it's interesting. We use tomato. So in tomato, most countries use TY1, TY resistant gene for breeding. And we also introduced the Deniscia tabasi resistant So if you introduce both resistant against insect and virus, maybe we can control the disease more better. So this was published this year recently. Okay. Recently, we are developing collaboration with South Korea because we are very close countries, and Yujin Kyo, I think he supervised visited here, Sukchan-D, and one of young scientists, he also got PhD in Sona University, SMU. So we are doing collaboration research to understand the diversity of TYCB in Japan and Korea. Okay. And also Taiwan is an important collaborator because they wild vegetable center, and Derek is a pepper breeder. And we are always discussing tightly because we want him, for example, him to apply our knowledge to actual breeding and help the farmers. And last month I visited Taichung. There is a nice university, NCHU, National University. There is also a big department of plant protection. So they are main biologists, some are focusing on Begomovirus. So we are now trying to develop the collaboration relationship. And also India, they suffer a big problem with Begomovirus. So I've been developing a network with mainly National Institute, Indian Council of Agricultural Research, ICAR, in New Delhi, and also Bangalore, a center for vegetable breeding. So here I would like to start the story of our science since I became a PI in 2011. So initially, I didn't focus on the Begomovirus. I was using peppers, but my supervisor gave me a topic related to temperature sensitiveness. So I took my PhD with this topic, but because this topic was, how to say, little difficult to make it larger and obtain the research funding, so I gradually shifted to different topics. First one is capsaicinoid biosynthesis. It's a chili hotness of peppers, and then gradually shifted to flavors of pepper fruits. We have published several nice papers, and then we are now mainly focusing to Begomovirus resistance. So because I'm a researcher in university, we have to make, how to say, positive growth cycle, not only research, but also education, because we have students. And when I initially started my career as a young scientist, I have nothing, no student, no facility, no money. So what we need is first publication. If you have good publication, we will have a good collaboration, research funding, and good students. So this positive cycle is the most important thing, and for that, good research topic is a critical thing. So I was thinking, what should I do as a PI? Because yesterday I was student, next day I become a teacher. I have to guide my student. So in Japan, when we cultivate peppers outside in the field, we normally transplant in April and finish our cultivation around October because temperature will be going down to cold for peppers. And when we back to Indonesia, my image was that because Indonesia is tropical country, you have good climate always for peppers, and you must have huge production. That was my initial thing. But when I came here, this is a photo took in Aceh. This was the next slide. There was disease symptom. So initially, I didn't know what the pathogen was, but when I checked the publication, it seemed to be Begomovirus, virus. So I further checked the publication, and there was no report related to Begomovirus resistance in peppers. So I found out, oh, this could be a good topic for me to become pioneer in this area. And because I prefer more applied research, I want to somehow contribute to actual agriculture production. So, Gemini can be divided into 14 genus now. Begomovirus is the biggest one because it now contains more than 445, almost 450 species. So that means most of the crops are affected by this virus. And Begomovirus can be divided to two types. One is bipartite, which has DNA A and B, two circular single-stranded DNA. And another one is monopartite, which only have DNA A accompanied sometimes with alpha or beta stabilizers. And both of them are causing big problems. And there are several photo crops, tomato, soybean, many types of crops are affected. And the difficult point is that this virus is transmitted by a small insect called whitefly. This is very small. And the basic management to control the disease is to treat insecticide or put some insect-proof net around the greenhouse. But most countries cultivate crops in outside field. So treating insecticide is not working so well. Also, putting the net is not ideal method. So we need virus resistant variety each crops. So there are several examples. First one is pepper. Pepper in Indonesia. Next one is tomato. This was documented in Spain. Also eggplant. This is the photo in Indonesia. And please go next slide. And we are also interested in cucumber, melon, squash, watermelon, cucurbit crops. So these are the target for my research. So our final goal is to solve this problem, but we are researchers in academic areas. So my mission is to prepare a tool for breeding. For that, we need to clone the resistant gene, prepare the DNA marker, so that mainly private company can do the breeding. And in Asia, this is an example of India. The situation is so complicated. There are so many DNA. These DNA of begomovirus infect only pepper. So they have like 14 or 15 species. And the combination is different DNA and something like that. So almost impossible to understand the diversity. But how to combat this virus is a big issue. So we divided our research to four stages. One is virus diagnosis in the field, collecting sample, and prepare the inoculation method, and do the screening of the resistant material, and do the identification of the resistant gene. So in the stage one, we did field research in Indonesia. Right? So as I explained previously, Indonesia, we have been collaborating with researchers in Banda Aceh, in the northeast of Sumatera. I'm recognizing Indonesia as one of the model field for begomovirus study. So what we did in the first experiment, research is very simple. Visit the actual farmer's field with my collaborator, collect sample, extract DNA, and amplify the circular begomoviral DNA, and then clone the plasmid to do the sequencing to understand the diversity of begomovirus. So when we draw a phylogenetic tree, these calculate one the isolated virus. So in this study, we visited 5 fields. Each field had like a 500 to 3,000 plants, and almost 100 plants are showing symptoms. And we randomly picked sample and sequence, and we found out that at least 3 virus species, 2 are bipartite, pepper CID, QICKV, and another monopartite one, AYDV, was infected in pepper. We continue these kinds of research, and we now know that pepper CID is a main predominant begomovirus species. And in 2017, we sampled pepper, tomato, lycocana, tobacco, and we recovered same virus species. This was very close to pepper CID, but a recombinant one. So we named this virus H virus pepper CAV. So these two viruses are model virus for our pepper study. Okay. And during those studies, we also sample other crops like tobacco, tomato, eggplant, squash, and cucumber to isolate the fluorescent viral genome. And in the next slide, we sequenced different virus species like TYHKV, YVV, TOHMDV, SLCCMV. So the main object is to understand which virus is the main target for resistant breeding in each crop species. And we also develop virus species specific primers to do the diagnosis more easily by PCR and electrophoresis. So in stage 1, we understood that which virus is a problem. So next, we want to develop inoculation method. Yes, which we use whitefly is possible, but you have to always take care of whitefly, manipulate, and because you use a foreign virus, it's less to use those in laboratory because if the whitefly having the virus goes out, that will be a big problem. So we chose different methods using agrobacterium. You learned in the lecture, agrobacterium is a bacteria to transform plants. And, in natural conditions, they may plant both in roots or stems. Okay. And agrobacteria also have plasmid. Is for PI plasmid, which are used for making transgenic plants. So our idea is not special. This has been done by many biologists. So if we put begomoviral DNA in PI plasmid, plants will be transformed by agrobacteria so that they can be infected by begomovirus. So we call these infectious forms. So in our case, virus was bipartite, which means they have DNA A and B. So initially, we cloned full length DNA and partial length in the first plasmid. In the second plasmid, we cloned DNA B, separate, and mix the agrobacterium and inoculate the plant. This can work for model plant like Lycocana, Benzamiana, or Tomo. But if we go to peppers, no infection. So what we did was to clone both DNA A, B, and B in the same plasmid so that we can use just one agrobacterium. And by doing this, when we inoculate agrobacteria, it will do the same thing as a natural condition. And it will show the disease symptom. So many methods are used. One is agro-infiltration. We put agrobacteria in buffer and penetrate the leaf or confidence. In other method, we culture the agrobacteria in solid LP plate and take a little amount of agrobacteria and put them on the hypocotyl using toothpick and pick with insect finish so that agrobacteria can go inside the stem to inoculate the virus. So we initially used Benzamiana and then moved to tomato. It took like 1 year because we were the beginners, but it worked. And if we repeat same thing in peppers, was little difficult. We have two tips. One is the body, the density of agrobacteria is critically important in peppers. Low density is important. 0.1, we have 12 plants infected out of 30 plants, but before this 1.0, only one plant are infected. And also the inoculation stage of plant is important. When we inoculate young plants, 75 are infected, but if we use old plants, much less. So these are the important conditions, and we repeated and repeated using the single pepper accession. And because we want to do the screening, we have to inoculate it to various pepper cultivars or accessions. So we try different cultivars, but in this case, no plants were infected by agroinfiltration. So we need a method. And what we did is that we first inoculate to our model line. We can easily inoculate in this one. So we take some branch as a scion and graft to many pepper genotypes. So by grafting, when the grafting is succeeded, 100% of plants can be infected by Begomovirus. So this is critically important for screening and genetic mapping. So we also developed a method for cucurbit, cucumber, melon, zucchini, squash. And for this study, we use T origin and BB as a model species. One is from Spain, one is from Indonesia. Sorry. Now in Europe, they already have this virus and supporting huge program in cucurbit. Our government is also worried about this virus because it already invaded China, Taiwan, in future, and Japan. So we have to prepare. Okay. So we want to move to screening and genetic mapping stage. Okay. So in case of capsicum, right? So capsicum can be divided to 5 domesticated species, and capsicum onion is the most important one. You also grow some capsicum frutescens, which you call Chande lauri. And pepper is very interesting because the hot one is used for spice, and the non-pungent, non-hot one is used for vegetable. We Japanese prefer the vegetable one. Maybe you prefer the spicy one, right? And both are very important crop. And when we take the production, world production of pepper, fresh and dried, is about 42 million tons. So it's big consumption. And 46% is produced in China, and followed by Mexico, Turkey, Indonesia, you are a big producer, and India. And we can say that almost 75% is produced in these 5 countries. And most of these countries are suffering the problem with Begomovirus. Okay. And there are several hotspots. One in Southeast Asia, South Asia, West Africa, Central and South America is a hotspot with pepper infecting Begomovirus. And after we started our research, other research team from maybe India or Mexico or Thailand have published resistant material in peppers. But those work are mainly conducted in the field condition. We did same approach in Indonesia, but it was very difficult because in the field, not only Begomovirus, other RNA virus, fungi, bacteria, they just water stress, heat stress, so it's too complicated. So we took different approach using infectious crop. So in our laboratory condition, in a small room, we can cultivate like 3,000 to 4,000 plants, which is air condition. And using small pot, we can finish every experiment in 1 to 1.5 months. So it's very short term. And using a single virus isolate, which make everything really simple. And we screen more or less 900 pepper sessions. And these two are the most promising material. BFF5. This is a local accession from Indonesia, which we found in Aceh, locally called perintis. And another one is PG11. The resistance was introduced from different domesticated pepper species, capsicum pubescens. So crossing capsicum annuum with pubescens is really difficult. Our collaborator in private company crossed like 1,000 flowers and just obtained one plant. And the progeny is PG11. So these two are the nice material. They are already published, so and the DNA markers information is also available. So I hope everyone use this for their purpose. And initially we did mapping using BFF5, and here you can see BFF5 resistant, BFF4 susceptible yellowing, and F1 is susceptible. So we say it is the recessive resistance. And in the resistant line, the viral DNA accumulation is very low, restricted. So we can say this line is resistant against at least two Begomovirus species. And we did sequencing. We call RAT-seq using two parents and F2 progeny, more or less 350 individuals. And we sequenced every individual and obtain the SNP in their data. And then we also did the whole genome sequencing of two parents to develop additional DNA markers and to do the fine mapping. Right? So here, this is the phenotype data out of 346. Most are susceptible, some are resistant. So these are the phenotype data, and if we combine the genotype data obtained by sequencing, we can draw this kind of figure. So there is 12 chromosomes in peppers, and there is single peak on chromosome 5. It means there is a resistant gene only on chromosome 5. So we named this recessive gene small pep1. And we further developed a DNA marker to find map. And in the end, we narrowed down to a small region, 34 kb, and only one gene was located. So this should be the resistant gene. So when we sequence this gene, mRNA and DNA, there was a single SNP here, just one difference, A to G. This is critically important because by this, nice intron is not right, but actually included in the protein one. And there is additional 28 amino acid, and this makes the function almost lost in this gene. And this gene was called Koeda, which is very famous because tomato TY5 gene is also encoded in Koeda. So we didn't expect, but pepper and tomato have same resistance mechanism. Okay. And we also did a reverse genetic analysis using virus-induced gene silencing. This is a method to downregulate the expression of specific messenger RNA. So in this case, we use a susceptible pepper line. When we inoculate begomovirus with PRB base vector, it's susceptible. But when we silence Koeda expression by pigs and inoculate begomovirus, it becomes resistant. So that means if plants use a function of Koeda, it becomes resistant against begomovirus. And we also develop DNA marker to detect the posture SNP. Here we can distinguish the genotype, resistant one, susceptible one, and also the F1 heterozygous, which is susceptible. So we can distinguish the phenotype in 100% without any mistake. And we did similar thing in PG11, so I will go first a little bit. This is a susceptible number 218, and graph is PG11 here. You will see almost no symptoms. So it's very, very strong resistance. Okay. And we did mapping. Here we can detect the gene from different chromosomes, not on chromosome 5, but on chromosome 7. So it means the different gene, and it is not recessive, dominant resistant. So F1 plant will become resistant. And we map this gene large PEP2 to area which contains 17 genes. So one of these genes should be the resistant gene. And then we check the annotation data, which gene encode what protein. So here there is a RNA-dependent RNA polymerase, which we call RDR. It's famous because tomato TY1, TY3 also encode RDR. So surprisingly, pepper and tomato have same mechanism. And when we compare the sequence of two lines, the susceptible one has multiple mutation, which will call loss of function. But this one has a functional. Okay, and when we check the original sequence of other capsicum onion and Chinese capsicum, they all have loss of function mutation. So it means only capsicum pubescent has a distant allele, which is useful for pepper breeding. And we develop a DNA marker also, so we can predict the phenotype by markers. And the bigs, same as which we did previously in BFF5 to conclude that RDR is actually the resistant gene. And last 2 months, we published another paper from plant disease. This is my final goal. We combine two genes, PEP1 and PEP2 with single pet plant to enhance the resistance against more wide variety of Begomovirus species. And in this study, we use 5 pepper lines, susceptible one, moderate tolerant one, BFF5, PG11. So these two are from Indonesia, these two are Mexican Begomovirus species. So BFF5 from Indonesia work well against Indonesian, but not against Mexican. But PG11 is working for all of them. But when we mix inoculum, 2 viruses from Indonesia or 2 viruses from Mexico, in case of PG11, they are all like a susceptible cultivar. And BFF5 is working still nicely. But if you go to the Mexican one, it's susceptible. So what we did is simple to combine 2 genes. This one has homozygous PEP1 and heterozygous PEP2. This one has homozygous for both genes. As you can see here, there is no symptom, even infected by 2 Mexican Begomovirus species. And DNA of virus is very low, so that we can say by combining 2 genes, we can almost completely control the disease in this case. So we have to test whether it's same in the field. So for this study, we need more collaborators and help from seed companies. We are trying to set up these. Okay. And because work in pepper and tomato, also eggplant will be taught by Nadya, so I will not introduce. The cucurbit is also an important one. And we have been screening cucumber and melon, both from 700 accessions. We have 5 good accession in cucumber, 1 good accession in melon. For example, melon. On the right-hand side is susceptible, but this number 198 is showing no symptom to mutated virus. Okay. And for cucumber, we have already published last year, and also RDR was contributing to the resistance. So same thing, same approach we did mapping. In this case, it was QTL, so resistance is controlled by 2 genes. One on chromosome 1, another one on chromosome 2. We clone the chromosome 1 gene by 5 mapping. And we check the annotation data, and there was RDR. So this goes next. We compare the sequence, develop the DNA marker, and next. We did bigs. Doing bigs in cucurbit is little difficult, but it's a good virus vector for cucurbit for LSD. So here, when we inoculate Begomovirus with empty X vector, it's still resistant. But when we silence RDR expression, the plant becomes susceptible. So it means RDR is also conferring resistance. Tomato, pepper, and cucumber. So this is current understanding. In tomato, the work is most progressed, mainly by US, Israel, Netherlands, and Japan. EY333A, Americ, they got encode RTR. EY5 is encoding pelota recessive gene. EY2 is encoding a receptor gene. And this year, EY6 was cloned as a 41, which is same with cassava. And we have cloned 2 genes from pepper, 1 gene from cucumber. We are now trying to map from 2 genes, additional 2 genes in cucumber, 2 genes from melon. And Nadya has already identified 1 dominant gene in eggplant. So this gene could be an important tool for breeding, but now we have modern techniques like gene editing or EMS mutation. So in this case, these recessive genes are very useful because if we break the function of the gene, it will gain. And also, if your government accepts transgenic plant, of course, we can use this dominant gene in different crop species. So we need to prepare more and more genes to help agriculture. So all of our publication is on our web or ResearchGate. So if you are interested, please access ResearchGate and you need to download PDF to know what we are doing. And this is for students. Now Nadya is doing Ph.D, master and Ph.D in support from Japanese government. We call M.Agr.Sc scholarship. This is a good scholarship. They give you salary per month, tuition, air ticket is prepared. So everything is prepared, and in maximum 5 years you get support. So if you wish to do master or PhD in our lab, if you wish to contact me, and there is a good opportunity for postdoc for JSPS funding agency. The salary is pretty good, maybe better than the young assistant professor. And through support, it can be obtained for 2 years. And also for senior experience professors, there is also invitation fellowship. One is 2 to 10 months, and another one is very short, but you will have enough support, financial support. So depending on your situation, if you are interested, please contact me. And because Nadya is finishing her PhD next March, I'm very much interested in inviting another new graduate student from, for example, yes. And of course, if you have budget by yourself or your support from your government or university, I'm always welcome to young people to stay in our lab. We have already welcomed from India. Just short stay will make you change a lot. 1 month is enough. You can see the different world and maybe find another interest. So if you have a chance, please also contact me. So this is my final slide. Most of the research was conducted by graduate and undergraduate students in Kindai University from Kyoto. And my partner in Syafira University is important. And also there is collaboration with the Japanese university, and especially the private companies are good collaborators, Taki or and some others. They always give me good information and help me to prepare the population like processing their professional, and they can apply our knowledge. Also, I want to thank the funding from Japanese government. Okay, so this is all. One more, just show. We are now preparing an international congress for horticultural science, IHC, next year in Kyoto. So, and in 2027, actually, Indonesia is preparing AHC, Asian Horticulture Congress. So we hope many researchers from Indonesia, horticulture, breeding science, biology, any topic is okay. So if you are interested, please join this congress. And let me go to the next slide. We are now preparing 27 symposia, and the 26th one, which was the next one, I'm doing the combiner with a professor from Australia and China. Okay, and this is a special symposium for Aichi 2026. We are focusing horticulture, East Asia, Southeast Asia, and Pacific. So basically any topic is okay. Fundamental, applied, biology is okay, anything is okay. So please feel free to join this congress, and if you have any help, please contact me. Thank you. So if you have a chance, I'm waiting in also in Johto. Thank you.
Okay, thank you. Okay. Thank you, Dr. Sota Koeda. Very interesting topics. I think you explained from the very basic problem, in the field, and then now you have a lot of data from your research. So it means that it's not very easy to solve the problem in the field. Sometimes when we see diseases, in the field, we just want to jump and then help the farmers to solve the problems, but we don't, understand the causal agent, the factors involving the development of the disease, something like that. But from your research, probably about 10 years now. Yeah. So you gain many information, very important information, and then hopefully it will end up with a good solution for the disease. Because Begomovirus, as you mentioned, is very important problem in our agriculture. Okay. If you have any question, just keep for a while because we are going to hear the second speaker. So please welcome Nadya, Syafira, Pohan.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. Thank you for having me in the Department of Plant Protection, IPD. So I am the first international student supervised by Dr. Sota Koeda and also first PhD. So today I am coming to Bogor to accompany him and also take for any opportunity working in Indonesia as a researcher. So today I'm going to talk about what I have been doing for the past 6 years. Approximately after I finished my undergraduate study, I directly got a chance to extend my master. And after I finished master, I got an opportunity to extend to PhD under the supervisor of Dr. Sota Koeda in Kindai University. So yeah, I am originally from North Sumatra, Medan, but I spent my undergraduate study in Aceh, the Syafira University, for approximately 5 years. And during that time, I met Dr. Sota Koeda, which is doing collaboration with my former supervisor, Professor Dr. Eli Kusumawati. And I get opportunity to interact with him and do research and then finish my undergraduate study. And then we decided to apply for a Japanese government scholarship. And I successfully finished my master. It's in the middle of Corona, but thank God I can depart to Japan safely and finish my master. And then right now, I'm a doctoral student, 3rd year. Next. So I think we have already catch the message from what is the concentration of the research led by Dr. Sota Koeda. And to summarize, actually, there are several point of strategies that we believe in our research should be applied to make our research output could be applicable for the society. First, we have to determine the causal agent of the begomovirus and how serious the damage is. So if you want to study begomovirus resistant gene, you have to determine what virus in your area, and then, of course, because we are studying agriculture, we have to see the damage of the production loss that could be affected by those begomovirus disease endemic. And then after we understand what happened, what virus is the causal agent, we go to the field with either identified begomovirus resistance material or gene to evaluate for multiple years to get the idea whether or not it could be sustainable, and especially whether or not that particular resistance material or gene could affect to mitigate the production of loss. Because what the farmer afraid after the production loss, right? And then after that, because I've been spending quite some time in the field, I right now for my PhD focusly on identifying begomovirus resistant gene, applying a molecular approach to find a gene in eggplant. So from that three strategies, actually I've been conducted one actual research for each strategies. So in the first point, we have to determine what is the pathogen, the causal agent. And in our case, we choose Aceh as a model field because I was there and I had the opportunity to in charge in the research. So in the first point, I'm going to talk about pepper yellow leaf curl Aceh virus. This is the novel recombinant virus from Aceh that we found in around 2017. Next. So initially, from the initial works that Dr. Eli Kusumawati and Dr. Koeda conducted, they found 80 to 100% pepper in the field, showing begomovirus typical symptoms, and the causal agent was these three viruses. It was a long time ago. But then in 2017, but the study was published in 2019, there is this new recombinant virus, which turned out to be a recombinant from the previously predominant virus, PEP-Y-LCIP. Maybe some of us has already know that PEP-Y-LCIP is the predominant virus in most areas in Indonesia. But in Aceh, we found one recombinant from IP with another virus. So what I have done in this research is I try to characterize what is the difference of the disease symptoms, infection effect, especially in the growth inhibition between plant infected by PEP-Y-LCIP, the parent, and PEP-Y-LCIP, the recombinant. So in the previous research, because we have constructed PEP-Y-LCIP and PEP-Y-LCIP, I tried to inoculate several Solanaceous crops with these two infectious clones and combine the development of symptoms and then the effect to the fresh weight, which represent the inhibition of growth. And then I isolate DNA to further see whether the accumulation of higher viral DNA is associated to the growth inhibition. So I try with model field, model plant, Nicotiana bentamiana, pepper, tomato, and one tomato which carries resistant genes TY1. So this is the result for my Nicotiana bentamiana, in which we can see that PEP-Y-LCIP is suppressing the growth of the model plant itself in comparison to the parents PEP-Y-LCIP. And we can see next that the cause of this higher virulence is because PEP-Y-LCIP can accumulate at least 3 times higher virus DNA in comparison to its parent PEP-Y-LCIP, and that higher virus replication associate with higher inhibitions of growth. So that is why the fresh weight of Nicotiana bentamiana is smaller than the PEP-Y-LCIP infected plants. Next. So we also care about what happens if these two viruses infect cultivated crops like pepper, tomato, which is also major plant crops in Indonesia. So we tried with pepper and tomato, which are susceptible. As you can see, Pepuai LCAV developed quite different characteristic of symptoms in comparison to its parent. Pepuai LCAV caused more like yellowing symptoms, but Pepuai LCAV did not cause a significant yellowing symptoms, but a significant distortion and also significant growth inhibition. And clearly we can see the tomato infected by Pepuai LCAV suppress the growth of the plant because the accumulation of Pepuai LCAV is higher than Pepuai LCAV. Next. So this is the one of the interesting thing because TY1 is widely used, especially in tomato crop production area like in southern Spain, in America. So we want to see whether this Pepuai LCAV, which is originated from Indonesia, Southeast Asia, can be mitigated by the TY1 itself, which encode RDL. So we try to inoculate Pepuai LCAV to tomato cultivar Momotaro peas from Japan, which TY gene is heterozygous. And then we can see that Pepuai LCAV infection did not cause any significant difference, especially in symptom development. You can clearly see that there is no symptoms in Pepuai LCAV infected plants. But when we infect with Pepuai LCAV, the plants getting smaller and then the symptoms getting significantly visible. And then why is that happen? Because Pepuai LCAV can accumulate higher DNA in Momotaro peas, TY1 tomato, that cause the suppression significantly on fresh weight of the plant. So next. So from this research, the point is that TY1 Begomovirus Resistance, which commercialized in many areas of the world, is not sufficient against our Begomovirus in Aceh. So maybe there is also similar case if we see recombinant in Java, in different islands of Indonesia. So this kind of study is very important to see whether what have been developed is enough for our endemic area. Next. So we also question with when Pepuai LCAV start to replace Pepuai LCIV, especially in Aceh. So we try to check the infection of Pepuai LCIV and Pepuai LCAV again from our samples, which we collected from number of field using species-specific primer because at the initial time of the research, we did not have a PCR primer that could distinguish the difference between Pepuai LCIV and Pepuai LCAV. So we tried once again. Next. And from the PCR detection, we can see from 2012 to 2016, here the green area, Pepuai LCIV was still predominant infecting species without infection of Pepuai LCIV or mixed infection between both viruses. But in 2017, 2018, 2019, infection of the parent Pepuai LCAV start to decrease, but the infection of Pepuai LCAV start to goes up more than 15%, and Pepuai LCIV only in fact mix with the recombinant virus Pepuai LCAV. So from this data, we can maybe see that how fast the Begomovirus recombinant could emerge in some endemic area in Indonesia. Just in 3 years, it can replace. And I think in 2022, after I finished my master research, Pepuai LCAV still become the predominant infecting species in Aceh. So this kind of monitoring is very important to give us a prediction whether or not Begomovirus species in Indonesia can change rapidly, so we can mitigate as soon as possible what integrated management is applicable in the field, what kind of resistant genes is enough for controlling this species of virus. Next. So after understanding the virus in the field, we try to study the performance of Begomovirus resistant gene in paper, which was recently mentioned by Dr. Sota Koeda, encoding protein capelota in paper. Next. So actually, I have conducted 3 years subsequent field evaluation of Perintis, which is the donor of FY1, with different combination. So in the first year, I cultivate Perintis with other cultivars, and then I cultivate Perintis with susceptible cultivar, and then finally I cultivate F2 population of Perintis and Kencana to see the association between genotype of the individuals in F2 to the phenotype, which represented by the disease severity index and also the production. But this time, because the limitation of the time, I'm going to talk about number 3. Next. So this is the condition, materials, and method that we applied in our 3rd year of field study. We actually conducted this research in Aceh right before I departed to Japan to start my master. So I have 1 year gap after I graduated. Then I spent 1 year to do this experiment. I collect data and I record one by one. So we use 420 F2 plants together with the parents as a control. And then we record the disease incident, disease severity index, and then to represent the yield data, we collect the data of the production. We also check the infection of the virus, which are 4 predominant infecting species, which is IP, P.P.Y.L.C. IP, P.P.Y.L.C. AP, T.Y.L.C. KP, and monoparta AYPP. And then we did genotype the locus of P.P.Y.1 in F2 individuals. Next. So the point of this research data, I would like to show you is that from here, so this is the F2 population, correlation between genotype and phenotype. Because P.P.Y.1 is a recessive resistant genes, of course, we expect the homozygote recessive allele to show resistance, to show low disease severity index. So this is the scale of the disease index. And then the other genotype is expected to be susceptible. So as you can see, next, as you can see, the homozygote recessive allele of P.P.Y.1 had the significantly lower number of disease severity, is also obtained in the parent as a control, which showed that the genotype and phenotype correlated for this F2 populated in the field. And because, of course, we in agriculture expect for high result, I also record the data of fruit, next, which shows the resistant allele of P.P.Y.1 had at least twice of that other susceptible genotype. So it means if we cultivate printing in the field and the plants get infected by begomoviruses, at least we can expect double production in comparison to the susceptible cultivars. Next. So that's why P.P.Y.1 is responsible for DSI and higher yield in the field. Next. So this is the visual representation of Perintis and then Kencana. It is not perfect. It still shows symptoms. But if you go to the field, farmer will directly tell you that we will cultivate Perintis because it can mitigate our loss of production. Next. Next. So first point of this research is that Printis is originated from Indonesia, and I think everybody can access if you go to Aceh directly. So it is potential for us to use this in combination, just like Dr. Sota Koeda has presented, with other Begomovirus resistant gene. And then because we have done this research in Aceh only, not in other parts of island in Indonesia, in which other begomoviruses may exist, different from Pepoi LCRP and Pepoi LCAP, we still expect to test Pepoi 1 resistance. So it's very important. Next. So after spending long time in the field, I tried to identify the Begomovirus resistance gene in eggplant as my PhD project. Next. Why I choose eggplant? First, I'm gonna introduce you. In Begomovirus resistance cultivar crops, actually tomato is the most advanced because there are numbers of genes have been identified, like this gene, and then this is the protein product. And pepper, recently, our team have also identified, but the product seems to be similar to that of tomato, you can see, and cucumber also same. In cassava, it's also same to that of TY6, DNA4D. But recently, there are increasing number of reports in begomovirus infecting in eggplant. But I could not find like significant reference, significant reports, which shows a progress on begomovirus resistance gene or identification study in eggplant despite of those damage reports. Next. So today there is no begomovirus resistant gene in eggplant. And because of that, we want to initiate this breeding program for begomovirus resistant gene in eggplant. Next. So because we have been spending so long in Aceh, and as I mentioned before, if we want to initiate the breeding for begomovirus resistant gene in any crop species, we have to choose our model virus, right? In Aceh, we have constructed these infectious clones because they are found to be infecting Solanaceae and also cucurbits plants in Aceh. And we have to determine which one of these viruses are our target as model because we have to inoculate. So we try inoculate these viruses, and next, only TY-LCKAP could infect and cause significant symptoms development. So in this case, we use TY-LCKAP for our model virus to breed for begomovirus resistance in eggplant. Next. So using TY-LCKAP, we actually in previous study have screened for about 736 accessions of eggplant by inoculating TY-LCKAP, and we found this number 820 is one highly resistant accession in comparison to the control. Next. So we further studied the inheritance of the trait in which we inoculate F1 progenies, and the F1 showed similar level of resistance to that of parent number 820, which tells us that this resistant trait is a dominant trait. Next. After studying the inheritance of the trait, we study the association between genotype and phenotype, in which we conduct RADSEC analysis using F2 population for 2 replication. Why 2? Because we want to confirm our result in the last. Simply, we conduct this composite interval mapping analysis, and you can see that there is a peak in chromosome 1, which means these resistant genes in eggplant is controlled by a gene located in chromosome 1. And this is the candidate gene inside the candidate region. First replication, second replication. Next. But after checking the list of those genes, we cannot see any protein product encoded by any ORF, which are correspond to the previous Begomovirus resistant gene, either in tomato, pepper, or any crop species. So it means it's a totally novel Begomovirus resistant gene. Next. So in this discussion, I'm going to show you, I have done these 3 major experiments. First, I conducted fine mapping, in which I replicate fine mapping 2 times to, delimit, to make it smaller from 700-ish kilobase pair to very small so that we can reduce the number of non-candidate genes. And then after that, we confirm that target region is the correct position where the gene is located. And then after we get the final number of the candidate genes, we start to silence reverse genetic analysis by virus-induced gene silencing of the candidate genes. Next. So simply the result of fine mapping and also the target confirmation. Next. 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 Looks like this. So I try to fine map from 700-ish here, and then I cultivate many number of, I think 96 as an initial, and then we decrease the candidate regions from 700 to 300-ish, and then after that, I recultivate like 1,250 F2 plants, and then screen by DNA marker, and then delimit again to 139 kilobase pair. And then whether or not that's the location, we cultivate F3, and then after that, we get final target region, 113 kilobase pair, which contains 10 genes as a candidate gene. So from 61 genes, we delimit to 10 genes. Next. So this is called eggplant yellow leaf curl disease virus resistance 1 or EY1 locus. Next. So there are 10 genes inside those regions. Which one is responsible for this resistance in number 820 eggplant? So first approach, we need to check every open reading frames of the gene, whether or not between the resistant and the susceptible accession, whether or not there is a non-synonymous mutations, the difference like substitution, deletion in amino acid in nucleotide, which also affect the amino acids. And then after that, we're gonna check by the mRNA expression, gene expression. And finally, next, we chose these 5 genes because there is a non-synonymous mutation. And number 6, there is a significant expression in both of the accessions, which may be responsible for this resistance. So we test these 5 genes. Next. By virus-induced gene silencing. So maybe some of us has already know. We try use the number 820 Begomovirus resistance accession, and then for the 5 target gene, we clone the gene, partial part of the gene, and then we ligate to the tobacco rattle virus, which is now to be effective for silencing gene in tomato, also pepper. But this time, we did not have any clue about eggplant, so we just tried an error. And then we did the inoculation mix between tobacco rattle virus, mix in one buffer together with TYLCKAP because we want to show the down regulation of each genes associated with resistance or susceptibility after the infections of TYLCKAP. Next. So this is the treatment. First, we need a negative control, positive control, and 5 target genes. Next. Next. So this is how the virus gene-induced silencing results look like. This is a positive control. This shows the silencing is started around 20-day post inoculation. This is the negative control. And all of these look similar, but next. This one is suspicious. So silencing the protein called NEN3-like lead to the susceptibility, while other did not lead to the susceptibility, which means these proteins might be responsible for Begomovirus resistant gene in number 820. Next. So we try to check whether or not the gene is actually downregulated or not. Next. And we found that, of course, all genes are downregulated except gene G, because maybe the expression is quite low, so we could not successfully silence that. But for the NEN3, it is silenced. Next. And next. What is significantly interesting is that under all the silencing treatment, only silencing the gene A, which encodes SMN3 proteins, could increase the accumulation of Py-LCK DNA, which lead to the susceptibility. Next. So the downregulation of SMN3 protein like increase allow more virus to replicate. Next. Next. So to study the structure of the gene, we check the open reading frame of SMN3 like encoding ORF, which you can see there is 3 minus base pair in number 8 to 0 in comparison to the susceptible line. And then, of course, it allows the lower number of amino acids. And next. And then after we check in SMN3 like encoding ORF in number 8 to 0, there are 5 non-synonymous mutations, 2, 3, 4, 5, and 6, and 1 deletion. Which one is affecting the function to the resistance, we still do not know. We cannot tell you that this deletion associated with this resistance rate, but maybe all of them are associated to the resistance. So we next try to find literature whether or not this SMN3-like protein have been studied previously by previous researcher. Unfortunately, only one study could be found. It is Arabidopsis NEN3, which is responsible for inoculation and morphogenesis. So it seems like this gene is encoding exonuclease protein, and the motif found in Arabidopsis thaliana is also similar to that what we found in eggplant. So probably the function is exonuclease, but we still do not know. Next. So that is the big prospect for how Begomovirus resistant gene encode different protein. Of course, rule different mechanism than any other identified Begomovirus resistant gene in vegetables. But the point, this is very important for the future breeding in vegetables. The homework is that because we conducted this Begomovirus resistant gene identification using Southeast Asian Py-LCK tomato yellow leaf curl Kanchanaburi virus, which are concentrated in Southeast Asia. Using EY1 is still unknown whether or not it can be effective against other Begomovirus species. For example, in Brazil, in American continent, there is tomato virus, which is significant for eggplant production, and then also in Africa, and then also in India, where it's much more complex. So we need to test this EY1 furthermore. Next. So it's a next challenge for the eggplant Begomovirus resistant gene study. Next. So I think that is all from my presentation. I would like to express my gratitude to my former supervisor, Dr. Eli Kusumawati and Professor Sabarudin in Koeda University, and also my current supervisor, Dr. Sota Koeda. And also many parties have been supporting me to conduct this long research, and my funding mostly coming from the Japanese government. And today, thank you for the audience. Hopefully, this could be a very valuable information for all of us. Any question, discussion, and yeah, you can ask me anything. Hopefully, one of you or many of you can follow my time to study abroad. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Okay, thank you, Nadya, for a very good share of your research. Oh, where are the students? Oh, they have classes. Okay, no problem. Now I think we can open for questions and discussion. Hopefully, this will improve our understanding about the Begomovirus. Any question maybe from the floor here? Bu Rita, any other? Rita, someone? Belum ada? Okay, you can start, Rita.
Thank you so much. It's very interesting presentation by Dr. Koeda and Nadya.
Ada
My question
banyak sih, Bu.
will be to Nadya.
Yang mana?
What is the approach when you
Tampilin aja nih.
doing the disease severity indexing using based on the genotyping? For the phenotyping, we
Harusnya ada.
usually use the symptomatic or based on the symptoms, but for the genotyping indexing, what's the approach that you use?
Bisa.
In each study I've conducted, especially in PEP-Y1 for the field, we try to document the PEP-Y1 genotype in F2 by using DNA markers, which detects SNPs between BA-PEP5, predicted in BA-PEP4, and then we subject to HRM, high resolution protein analysis, using quantitative PCR, qPCR. But one choice, if you don't have the facilities, you can also use like simple indel markers, like you do PCR and then you do agarose technique, which can distinguish between whether or not your target accession contains PEP-Y1 or not.
Oh, okay, thank you.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
My name is Rita. What is your Ritalis? Konnichiwa. This is an interesting topic. I started one question. Why use agrobacterium for inoculation paper yellow blister virus? Why you use agrobacterium? Thank you.
Really good question. Several reasons. In natural, the white bright insect is transmitting the virus. So, yeah, if we have enough support, I mean, if we use white fly, we have to manipulate the white fly to several thousand always and use it for experiment. So that is part of building greenhouses, facility technicians, and also we are using the foreign Begovirus, not in Japan. So if any of the white fly having the Indonesian Begovirus go out from laboratory condition to our farming field, we cannot stop the virus, right? So that will be a problem. So it's one reason, and because Begovirus is DNA virus, it's very easy to prepare infectious clones with bacteria. And we can inoculate a pure virus isolate. So these are the main reasons that we use agrobacteria. And actually, there are other methods like particle bombardment, which we use DNA gun. But that facility is
Mungkin saya panggil
expensive
aja ya.
and
Itu
you need
kalau mau
both.
nanya langsung ya.
Initially, I was
Kita-
just scientist and didn't have enough money, so I chose the base. So those are the reasons.
Okay, is there any more question from the people here in the room? Okay,
Okay, thank you. I want to ask a question for Nadya. Your research is very interesting because in Indonesia, especially in Java, eggplant is only planted near the chili, so it becomes the host and also the source of inoculum as well as the vector for the viruses. And I want to ask you about how to get the single virus for inoculum, either PYLCAV or PYLCAV, because sometimes many kind of viruses can infect the same plants. And how can we get a single inoculum, the exact inoculum for our researchers? Because as I see on your PowerPoint since 2019, it is so hard, maybe 0% PYLCAV, and most of them are double infections between two of two of the virus. So that's my question. Thank you.
Thank you very much for the question. So actually, it was conducted by previous members. But I think one of our approach that has been a long time applied is that we collect the DNA of infected plants, especially like we're studying eggplant, right? In eggplant, infected, there's a symptoms, right? So from that symptomatic, we detect first by, of course, universal primers. And then after that, we did not know the virus. So in that case, we're gonna conduct RCA. So to check how, yes, is there any begomovirus or not? So we started to if we do not know the virus, then we start RCA, and after that we check the infected virus by sequencing and something like that. But because from the previous year we have the specific primer, at least to detect for virus, we usually just try to detect whether or not requires LP, requires AP, P1 or AYTP first. So if there is no infections from those four, maybe it's different virus, and we have to do the RCA and isolate new virus maybe. So that's when we determine, oh, this is one of our target virus. Maybe after like several years or something. Thank you.
Yeah.
Oh, yeah.
A little bit.
Yeah.
Your question was very interesting, and for example, by CIP and ABs mixed infecting the single plant, we will recover the both sequence. So initially we don't know which one belongs to IV, another one belongs to AB. So when we compare the fluent sequence, especially the intergenic region, is concerned between DNA and B. So comparing sequence, we can distinguish if A and this B is a pair. Another A and B is different. By doing this, we are looking at it by CSP. And C main range.
Very nice. Okay. All right. I think we have some questions from the audience in the Zoom. I'd like to highlight the last one from Dede Yusuf Ziaul Haq. I think this is the same question as I just mentioned. So he asked, I'd like to ask a question in advance regarding begomovirus isolation. Is it possible to obtain single pure begomovirus isolate? Right? This is the same thing without using agro-infectious clones. That will be a challenge. Yeah. In many cases, I noticed a mixture of viruses, and I wonder how we might purify the virus to obtain a single isolate for screening without agro-inoculation. I think this is the situation in Indonesia, because to develop infectious clones it is still difficult. So we rely on the bioassay, yeah, inoculation using wildlife, something like that. So, do you have any suggestion or recommendation how we can do the problem like this?
So actually, I have not that, but my problem is has this approach. What they did is initially they prepare mixed infected ground and treat healthy Wi-Fi and transmit to many plants, and they do the diagnosis and find the single infected one. So that if you keep this plant, you can use the inoculation source.
Okay. I think that's a good idea, but you really need a lot to have a single isolate infecting your plant. But yeah, it's a good try. Is Dede, are you still in the Zoom? Pak Dede?
Ada sih Bu,
Ada ya?
cuman di mute.
Maybe, yeah, okay. I hope the answer helped you to solve the problem. Okay, we go to the first question for Dr. Koeda. My name is Cristianto from Plant Breeding and Biotechnology Program. I think this is from IPB also. I would like to ask about the discovery of a SNP in peptide gene that is located in intron area that could distinguish between the resistance and susceptible chili pepper genotype. Besides having a function as a selectable marker, what is another function of this SNP gene? Is it possible that the SNP is having a role in the gene expression? Pak Cristianto, are you there? You can ask directly if you are there.
Kristianto siapa? Nugroho ya?
Cristianto Nugroho.
Ada Bu, cuman off cam
Mute.
dan mute ya.
So please.
Okay, so if I am understanding the question properly, so how this SNP affect the main mechanism? One is that because this SNP may pepper not to splice the nice neutron. They do not cut out the nice neutron from messenger RNA. So it will be made and transformed to protein sequence. And this will decrease. I mean, we are now analyzing that this will not cause complete loss of function, decrease of function as a delta. And related to gene expression, normally if plants have loss of function or decreased function proteins, gene expression will down. So maybe because of this, BFF5 have lower gene expression compared to the susceptible pair. So maybe this is the overall I can do right now.
Thank you for the answer. The next one is from Nurfianto Giar Pangidung from Malang.
Ada Bu.
Yeah, Pak Nurfianto, do you want to ask directly? Please active your speaker, maybe, if possible.
Mute Bu dia.
Huh?
Nggak ada. Ada cuman nggak
Yeah.
bereaksi
Okay.
apa-apa.
Then I will read the question from Nurfianto to Professor Koeda. What are the breeding and seed production strategies for chili varieties resistant to virus and aphid? Maybe he means the whitefly instead of aphid as a factor. Given that chili varieties in Indonesia are developed as hybrid and open pollinated varieties and adapted to specific geographic region.
Thank you for the question. So the breeding strategy differ from the recessive gene or dominant gene. So normally, I think more and more F1 hybrid carnivores are available in the commercial. So in case of F1 hybrid, when we are using recessive gene, we have to introduce the wisdom gene in parent A and B in homozygous state and cross them to obtain F1B. But in case of dominant gene, you just introduce wisdom gene in one parent as a homozygous state, and another one do not need wisdom gene. And if you make a F1, it will have the wisdom gene in heterozygous state, and those will show wisdom. So it depends on the gene.
Okay, hopefully this will answer your questions, Pak Nurfianto. Ada lagi? Maybe we have two more questions.
Saya tadi terakhir yang ini paling Bu.
Orang yang beda aja gitu.
Ini masih sama ya?
Itu Dika ya.
Mm-mm.
Question from Dika. Why use agrobacterium inoculation instead of whitefly? I think this is already answered. Is there any benefit? Yes, I think you answered that. Usually in Indonesia, the plant is attacked with several viruses at a time. Why we need to inoculate with only one virus? Okay, maybe you can address that.
Okay, that's a good question. So, in initial stage, we have to make everything simple. If you use 2 or 3 viruses, it's too hard. So we use just one virus, identify the wisdom gene, and after growing the gene, we did mix inoculation, addition, and show that it actually functions when you combine 2 genes. So important thing is to go step by step. And yes, at least in papers, and also it's not published yet, but also in tomato, combining at least 2 genes virus resistant gene is very functional. This is mainly because they have different resistant mechanisms. One is maybe RDR, one is Pelota or other mechanism, so that it makes plant more resilient against viruses.
Okay, I think that's the answer. Ada lagi kan? Nandung ya? Oke Pak Nandung apa sih? I asked more about the peppers resistant to Begomovirus. After we tested the PEPY1 gene using a molecular marker and a natural field condition test in our breeding materials, many lines have the PEPY1 gene. However, the incidence of PEPY-LCIV varies, ranging from 10 to 54% across that stage. What should we do next? And what source do you use to get PEPY2 gene?
He or she must be a professional. It's a really good question. So, first question, not only peppers, but crops, the genetic background also affects the resistance. So, although one cultivar and another cultivar has same resistant gene, the genetic background, it differs, the performance also differs. So, it's a 10 to 54%, maybe genetic background of specific pepper is more better in combination with PEPY1. And also in the field, the condition is always fluctuating. These are environmental factors also affecting the phenotype. And for PEPY2, unfortunately, this material now belongs to a seed company called Taki, and they have the material, but we didn't get any international patent, so basically it's not protected. And because we have already published, we have many contacts from seed companies, and actually like a big company like Syngenta also already have negotiated with Turkey for the use of PEPY2 with paying royalty. So in few years, I'm now thinking that the PEPY2 and PEPY1 contained material will be released in India
market and gradually used in different countries. And because I cannot say directly this, but it's not protected by patent, you can buy the seed and use it for breeding source. So maybe you have to wait several years, but in that time, not only PET1, but PET1 will be available. So I hope seed companies, not only in Indonesia, but other countries get interest and use it with our DNA marker.
Okay. Kayaknya udah nggak ada pertanyaannya. Alright. Do you have any more to say or to, to conclude our discussion today, or that's enough?
Yeah, the last question was very nice. If we want to apply our knowledge, academic knowledge structure agriculture, we will face really difficulties. But we have to solve one by one. And for that, the collaboration is especially important. Maybe between university and institutes, also seed companies, and the communication is always needed. So I'm always open. So please contact me if you have any. And also, I'm waiting for talented students. So please contact me or please contact me. Thank you.
Alright, thank you very much, Dr. Sota Koeda. It's been a very good discussion. And we are very honored to have the team from Kindai University, not only for sharing your research data, but also your invitation to join your research team. Hopefully, some potential students in this room or in the Zoom will, would like to join the team in Kindai. And as the last thing, we would like to present the certificate from the our Department of Plant Protection. We are very honored and very happy to have you here, especially for the students. I think they learn a lot from the research that you've been done, and hopefully it will motivate them to design a good research program for their stuff. Okay, can you please stand up in front, both of you? This is for Dr. Sota Koeda. Oh, Nadya, you have to. Okay, and for Miss Nadya Syafira, hope for your good future as a researcher. Okay. Thank you. Some more souvenir for you to bring to Japan. Let's see. Udah mau foto bareng semua? Iya, siapa? Rita DS. So I use master in biology, Uri, master or Ph.D. now? Oh, Ph.D. student. Also in biology, Musna is master in biology. And the four of them is in new students in plant pathology. They have not decided where to go. Ya, jangan numpuk di sini.
Squeeze.
Belum. Kenapa?
Ada nggak foto yang tadi bisa dikirim ke saya?
Paling saya backup dulu ya ke Google Drive paling.
Apanya? Mau minta foto.
Yang lagi presentasi atau yang ramean tadi?
Yang ramean.
Ada di ibu yang itu. Tadi ada pakai HP itu. Ah. Oh ya, oke oke.
Yang tadi.
Oke oke.